a fully functional software for medicinal and computational chemists called Synspace, which has been designed to avoid any need for cheminformatic skills for chemists or synthetic skills for computational chemists. We also have a end-to-end AI-assisted lead discovery platform called Aid platform, which includes the design components of Synspace, and it's fully integrated with some other tools, which I will briefly show. So lead optimization is being recognized as one of the most time-consuming and costly part of the discovery paradigm. It's been published by several authors that over 30% of the cost is spent at this stage, including the unsuccessful projects. But it takes somewhere between 2 to 4 years. Big pharma spend a lot of FTE months. Smaller companies try to get this done from lesser resources, but it is still true that it's a long and costly process. It also fails in one company and it succeeds in others. So it's not a systematic process which just sort of works everywhere. So the reason for this is obviously that multi-parameter optimization is a very complex process. It requires a lot of DMTA cycles, and we can fail at any point due to tox and other reasons. So this is a really challenging part of discovery, of course. So the key considerations impacting cost and timeline are the number and the number of cycles and the cycle time of this DMTA process. Synthesis in the design, make, test, analyze cycle is far the longest and most expensive. And of course, not including in vivo animal studies at the preclinical stage. But overall, the process is really burdened by the time and the cost of synthesis. And the cycle of a synthetic process can be anywhere between 2 weeks to 4 months, or maybe even longer for some compounds, because there will be molecules almost in every cycle which will be slow to make. Stragglers will come back with that data maybe months later, disrupting the design process because the information is missing. So this can really cause a significant issue in the design process that we don't have data for compounds. Also, there is a major mindset problem in this. Obviously, if you work with synthetically not feasible compounds, but they sort of present a compelling design idea, it's very hard to dismiss them and not start synthetic process. Once the synthesis has begun, it is really hard to drop a compound because the synthesis is failing. There's always another route to try, and therefore compounds keep sort of being pursued, and money is being spent, and time is ongoing. And lastly, we have a vast collection of virtual and commercial compounds available to us these days. E-molecules, real database zinc, they are all available. However, they only represent a very tiny fraction of a chemical space, and even though real database may contain billions of compounds. So they have really little impact after lead finding. We need to evaluate the chemical space around our leads with custom design tools, and that's why we have Synspace and our other tools for lead discovery. And lastly, the synthetic feasibility is a very important subject in everything we do. Chempass has built technologies to design and evaluate compounds that are sort of Teflon coated with synthetic feasibility. There has been publications by us and others regarding this topic, commenting on the synthetic feasibility of generative design by deep networks, also simple enumeration tools, and the scoring tools which we have sort of as a predictive synthetic feasibility predictors, which actually do a very poor job at predicting time to powder, if you will, which is the most important aspect in a lead optimization. How much time do we need to get a compound into an assay? So these tools are are really not vetted, and they are not a good evaluator of synthetic feasibility. So if we don't use the concept of synthesis in the design, then we will need to do full retrosynthetic analysis on every compound we design or want to seriously evaluate, which is a very costly and even insurmountable job with even today's computational tools. So Synspace is our software tool, which is really a comprehensive suite of different capabilities. As you can see on this screen, we have different modules for different tasks. And I will have a chance to show you a couple of these today, but really the key competency is scaffold design. We have multiple tools. The simplest one being OneClick scaffold design, which is I'll show you briefly, but we do have some other tools as well. We do have two generative design tools, which I will show you briefly. Retrosynthesis, a industry-leading library enumeration tool, or focused library design tool, I should say, multi-step, multi-library, multi-site enumeration tool, and some other design tools for libraries, which I will skip today. So how do we make this user-friendly? I think it's a very important concept, how to design by a chemist when you don't want to work with the cheminformatics. You don't want to maybe create substructure searches, which are very difficult to write. A good comprehensive substructure search tool is beyond the capability of most chemists. So OneClick Design was created to accomplish that, which only need the structure of a potential active molecule or lead, or a structure of interest for whatever reason. And just answering a couple of, selecting the scaffold you want to optimize, a couple of simple answers, whether you want to change the hydrogen bonding features, aromaticity, or ring size, or do you want to make maybe a monocyclic ring rigidified into a bicyclic. You can just click start and you get a list of scaffolds shown in the middle with properties, different rankings based on synthetic feasibility, building block availability, and properties, and other physical chemical properties and practical properties like cost, for example. So you get a list of suggestions, and separately you get a list of syntheses for all of them. In a separate tab, if a molecule has 200, then you get a list of 200. If it has 4,000, you get 4,000. But it's an easy, exportable and reviewable presentation. The library design tool is extremely powerful. It can actually do multi-step, up to 3 steps currently, but it will be unlimited in the next version. You can actually select multiple reactions by just simple drag and drop reactions into buckets, and then you can do a library design using that in your custom building block set or in a molecule building block set. If you want to do, you can do commercial availability or patent screening within Synspace, built-in databases. So we can tell you if SureCanBL has something similar in there, in that molecule you're selecting, and there's a lot of substructure search and similarity tools as well to sort of select the right compounds for further evaluation. We have, as I mentioned, 2 generative design tools in Synspace, which are very important these days. There's a building block base, so only requires sort of pieces of the molecule which you want to put together, and then the software can create a generative library. All molecules will have synthetic schemes and all other reagent data as well. And we have derivatization design, which we recently published as a new concept, which is a fully comprehensive and extremely user-friendly tool to manage really a full evaluation of scaffold and lead analog space around molecules. Each of them can be simply accessed from within Synspace using the built-in tools, which I will show you via a brief demonstration right now. But I think in this slide, I just wanted to demonstrate sort of the capabilities within Synspace of what somebody can do, for example, starting from a hit structure. So let's assume we have a hit looking like this. Obviously, one can do retrosynthesis, a full analysis, or one can do scaffold hopping, rigidification, as I said, or we can do just the generative design right away from the original hit structure, or multiple structures, of course. Once we have new scaffold ideas, we can do full sidechain design or evaluation of the sidechains at selective the different points one by one, or we can do a full generative design as well on the new scaffold. So I will try to leave this presentation now, okay? And to show you Synspace and give you a little bit of a taste how this works. Of course, we have all these modules on the top. It's a little bit colorful to be able to distinguish all these different modules via simple colors. And let's go back to our sort of demonstration molecule, our initial hit structures. So of course we can do retrosynthetic analysis. This is a relatively simple molecule. And you can see the software finds over 4,000 synthetic solutions to this compound. We can review these synthetic schemes one by one. You can see all the 4,000. You can view, export as a whole or just one by one. But I think you can copy smiles one by one and so forth. So there's a lot of features built in. We can actually review structures via schemes via two different mechanisms. In terms of working with the results, one can just select a scheme you like. Let's assume I want to evaluate the synthetic scheme, and I can initiate a derivatization design right from this scheme. The software will create a design scheme automatically for this molecule using that synthetic route. You have the different steps, the required building blocks. All I need to do is add a couple of filters if I like, one by one. You can actually add new reactions if you like. You can create your own reactions within Synspace. But everything is fully set up. All you need to do is just press start, and you can start a generative design. We're comparing to our target molecule via different similarity settings. This is fully described in our publication this January, which was released this January. But I think it shows you how fully automated the setup is, and you only need to work with the results later. But going back to our molecule, we can also do, of course, scaffold hopping. There are multiple ways of doing that, but the simplest is OneClick Design. And really, this shows you the setup of OneClick Design. I'm just going to duplicate this very, very quickly just to show you how it's done. You draw your lead, you select the scaffold, as I said, and if you wanted to keep the hydrogen bonding of a hydrogen bond acceptor or donor, you can mark it. Change aromaticity, yes or no. Change ring sizes or closed rings, and different stringency settings for the 3D overlap analysis and the hydrogen bonding. And just you press start, that's it. It's extremely simple, no knowledge of synthesis or other cheminformatics is required. And you get a list of scaffolds, as I mentioned earlier, with all the data and the synthetic schemes on a separate tab. Of course, I could just start a derivatization design with the same methodology for the scaffold now. So you can do the scaffold hopping and generative design very simply within the tool in a very straightforward manner. But you can also start library design. You can select multiple scaffolds which you like, which are, of course, come with synthetic schemes. I can generate a focus library by just giving it a new project name. Software will transfer the scaffolds. I can now pick reactions. From the synthetic scheme on the previous slide, I know that Grignard reaction, for example, is useful here. You can just drag and drop Grignard reactions. Of course, you can add other ones if you like, multiple ones. And you're set up with reactions. You can use the built-in or your provided building block sets, or you can create custom building block sets for each reaction step using the built-in tool. It will take you to a substructure search tool within Synspace, where I just say Grignard reaction. I want to get all the building blocks for Grignard. You can filter this for similarity or other tools if you like, and the software will transfer back the results to the design page, and you can initiate the design with the custom building blocks. So this is just a simple demonstration of some of the tools which we have in here, and how you can interact with the different modules within Synspace with these. And of course, this is sort of an independent tool which anybody can use in-house, or we can host and work with our partners like such. So I'm going to jump back to the presentation quickly and show you our the results for a case study on DDR1, which we published, as I said, where we compared the rivetization design, which I just showed you how to set up, to a GAN-based generative design published a year before in Nature. So this is an interesting project because they actually released all the 30,000 molecules designed by GAN, so we could compare directly to that from the rivetization design products. So GAN was trained by 1,400, a very large set of known actives, and we did derivatization design from 8 different ligands using the tool which I just showed you. We did some filtration of both sets. It's worth noting that deep network-based design will fail much more often, much more percentage-wise, for simple physical, chemical, and other unwanted filters. And then we would dock here over around 6,500 compounds to 3 different DDR1 protein forms: inactive, DFG in, or DFG out. These 3 different forms. And we looked at glide hit rates as an enrichment tool, as well as some selected statistics for the best top 50 and the best compounds. We also looked at, with scaffold analysis tools and some other visual interpretation, sort of the new features, completely novel findings by deep learning or by derivatization design, and the synthetic feasibility, of course. So what we can say is, doing these studies, that the glide hit rate or enrichment shows a much lower hit rate for deep learning. There is a much higher sampled chemical space, which does nothing in terms of generating hits. So they are sort of wasted designs, especially considering the very large training set for the tool. Here we are highlighting in magenta our designs, and in yellow the deep learning results in this plot. And you can see that there's a very large space in the middle, which is mostly sampled by deep learning only. When you look at the density of the structures, you can see actually that this is the most dense area. And when you look at the actual hits by a glide enrichment, you see that actually there are very few hits in the middle. So I think this is one of the reasons why the hit rate is rather low using the generative deep network tool. We are much more comfortable, obviously, with the 35% hit rate. And you could say that, well, one good hit is enough. But this is really not true, because you have a lot of false positive and false negatives in all these enrichments. So actually, you need a good number of well-represented hits in your design set. Now, we were also looking at whether we can reproduce prior art using derivatization design, and we were very happy to see that all prior arts known at the time by other groups were successfully reproduced, and we could design scaffold hubs within a single cycle using privatization designs, and they are represented here, and they are both published. So I think it's very important to know that this tool can be used very early in the optimization. So you just need one hit known active. You don't need a large set of training set, which you need for deep learning. We get better hit rate, better docking scores. We can do scaffold hopping and fine optimization with the same tool, and we can actually do that at the same time. And most importantly, the wet lab success rate for our tool is over 85% for 3 to 6 steps. And that's very important because most people would accept that from a single step sort of virtual design. So I think it shows that the design component which we do is really helps the synthetic success rate. Here's a separate POC. So that has to do with cycle time. The coverage within one cycle, and also the cycle time, because we can get compounds made quickly and we can cover a nice chunk of chemical space. But what about cycle count? Because both are important. So we are demonstrating that with a separate study, which is much more suitable for that. So we were comparing designs to the Nature published set by Hopkins in 2012, which was really a key publication at the time and since, showing that polypharmacological profiles can be designed by virtual computer-generated structures in a straightforward manner. And there are 6 different series here with different polypharmacological profiles, starting from the same starting molecule. You can see the number of cycles it took the authors to get to their sort of differently profiled target structure. So we reproduced the same study using the privatization design. And you can see that 3 of the 6 molecules was reached in iteration 2, 1 in iteration 3, and 2 in iteration 6. And these are the original cycle counts from the paper. So in overall, we could reduce the cycle count by 50% compared to this published data. So these design tools have been incorporated via APIs into our end-to-end platform, as I said, 8 platform, which has both of these design tools and some other ones. We do have docking analysis tools, DL and ML models, and other factors to cover an end-to-end discovery platform. What does these other tools do? And I think it's very important that, so of course we can design molecules which are synthetically feasible. And I didn't mention didn't talk about the technology behind the synthetic feasibility evaluation, and I would be happy to help anybody offline. Unfortunately, I don't have the time today to do that. But I think it's very important that we have some other tools which helps this process. We use active learning and automated analysis of docking results. So active learning can help us really enhance our throughput. With just a few % of the structures, we can get most of the actives. And since we have groups of actives in our design tools, we don't lose any of these sort of chemotypes in this process, but we don't need to dock, so we can dock hundreds of thousands of compounds very, very quickly with active learning-based tools. We spend a lot of time developing tools that can eliminate false positives in docking. I'm mentioning a couple of mechanisms here. This is a paper from others trying to do something similar just for the hydrogen bonding analysis of docking results. But we have all these different aspects built into our workflow so we can automatically remove false positives. And I can say that a very large number of poses get flagged or removed, as you can see, after these tools. And I'm showing you just one molecule with a very high, very low docking score, which is good. showing you how distortion of aromatic planarity was not penalized by docking. And obviously this structure is flagged or removed because it's not a good pose due to these strains. In terms of optimization by help by computational tools and models, I have just one slide here showing you that we have an active learning based optimization technique, and we use desirability scores for compound selection for synthesis. Again, very briefly, just to show you a case study where we compare in RED-R tool to a published data set by genetic algorithm from 2011. The performance, you can see that we get much higher IC50s from our active learning tool. And in green, you can see actually the performance of a group of medicinal chemists, which you can see that is better than genetic algorithm at the end, but worse than the active learning tool. In a different presentation format of this result is shown here, where you start from a very small SAR data set, which looks like this. Here are the PIC50s by color. This is what the genetic algorithm accomplishes over 10 cycles. You can see that it doesn't reach compounds in the more active ones in the exploration. This is what medicinal chemist 1 and medicinal chemist 2 accomplishes. You can see that they both do better a little bit than the genetic algorithm, but they both do significantly worse than our active learning tool, as you can see in the middle. And this is just a plot showing you a sort of a compound selection of a Pareto front of 3 different axes, one being activity, the second being an MPO score, and the third being diversity. And of course, compounds. These are compound sets actually in a marked red, which are sort of the best ones. And we have machine learning and deep learning tools, sort of models for different properties in-house, ready to be deployed as part of the optimization workflow. So just a couple of quick case studies at the end. I got maybe 2-3 minutes. One, just to show you that we can use our software tools and the platform at any stage. This is a late stage support case study where HERG issue showed up for our collaborator, and we had to help solve this. We used a number of techniques from our platform, and we were able to redesign this to a much more HERG-friendly series very, very quickly, within 1 month actually, for this partner. A second one showing you, it's a very interesting study I selected because it involves 2 steps. One step was a fragment design, and the second one is a library design for lead series. So this was a study where there's one key binding motif on all known structures in a relatively large binding site. So first we designed fragments for this highly conserved motif. And then in a second round and the third round, in 2 subsequent cycles, we designed different series filling this pocket in different ways, and overall coming up with 15 different lead series in a short amount of time. And the way it's done really is we're using all these tools in a sequential manner in cycles, of course. But I just wanted to highlight is that it was very important that in the first pass, we get warheads which meet the criteria of this tight binding into this very important part of the pocket. But all these warheads now multiple chemical handles. So we can automatically and very quickly design molecules using these handles that will be directed by probe vectors and pocket feature analysis in this pocket to come up with different series. And you can see that this is just one of these warheads and an overlay of all these different highly ranked structures which fill this pocket. This is series 1, and this is series 2, completely differently oriented in the same pocket. And we collaborate with our partners, of course, via very simple reporting tools easily understood by medicinal chemists or computational chemists with different rankings and other tools provided for selection. I'm going to skip this one. Just show you one last case study, which we have done on lead expansion and lead data analysis part of the project early. This you could also look at this as sort of a lead generation or patent busting example where there's one chemotype in the literature. We followed 3 MPO parameters, and this was a very conserved, tight chemical space covered by 3 different companies. And in 5 months, we've generated over 35 novel series using our scaffold hopping and generative tools with superior scores. Over 30 of these were selected for synthesis, about 3 compounds on average per series. So these are not libraries, these are cherry-picked molecules selected for model building or for potency explorations. And a very large percent was made very quickly, and there were a lot of interesting compounds coming out of this. So I'm out of time, but I would be happy to further elaborate on any of these, as well as our design technology here offline or as part of the networking within the conference. And I think I just want to close my talk thanking you for your interest. And with this slide, you know, I want to show you that we have a very flexible collaboration models, and we would happy to work with anybody on a very different flexible arrangements, including financially contributing to a project if you find it very compelling. And thank you. If there are any questions, I'll be happy to answer them.
Greg, actually, we have a question for you. One of our attendees has asked you a question. So, do you mostly do docking when you have a hit and use Synspace for optimization of the compound?
As a first pass, yes. We mostly do first selection is done by docking because depending on the project, we can generate anywhere between 2,000 to 1 million compounds in a relatively straightforward manner. So that's a first good enrichment tool, especially if we have large number of compounds. But you can apply to any any tools for sort of compound selection or ranking or analysis at this point. Synspace or our platform will allow a full export, and you have all the synthetic and building block data in the export in an SD file, which you can connect to any database and just apply other tools as you wish.
Okay, so great. So do you also do structure-based drug design?
No, we don't develop tools for structure-based drug design specifically. We use commercial tools for that part, and those are built into our platform. So we can work with a lot of different tools if you have preference, but we do have a couple of them preferred by us. But they are incorporated into our platform, and we can just exchange them if you like to have a different tools.
Okay, that's perfect. So, Antonio, thank you so much, Greg, for your presentation and for such an insightful knowledge. So, our audience, if they will be having any question, I will ask them to ask you questions on this main stage chat. Okay. Thank you, Greg. Have a great day.
Bye bye. Thanks.
Bye. Okay, so ladies and gentlemen, our next speaker is Robert Soliva. So, and we are sorry for the inconvenience, and Antonio Gomez will complete his rest of the session at 1:30. So, if you want to attend his session, you just need to wait for 1:30, and he will be with us at 1:30. And now we will be connecting with Robert Soliva. He's from Horizon Genomics. Hello, Robert. How are you?
Hi, I'm fine, thank you. Can you hear me okay?
Great. Yes, I can hear you.
Okay, because there's no way for me to know whether this is working or not. So this is
yes, it's working.
It's working. Okay. Yeah. Okay.
Yes. So Robert, you just need to share your screen. Can you see the monitor-like icon?
Hold on, please.
Okay, sure.
Yes. Let me see if it works.
Okay. Yes, great. I can see your screen now.
Hopefully, you should be seeing my first slide. Is that correct?
Yes, yes, I can see it. So, ladies and gentlemen, let's welcome Robert Soliva, who is working as head of drug discovery at Horizon Genomics. So, Robert, you can start your presentation now, and the stage is all yours.
Okay, thank you. So, hi everyone. My name's Robert Soliva. I'm currently head of drug discovery here at Horizon Genomics. First of all, I would like to thank the organizers, the Drug Discovery Summit, for this kind of invitation. It's kind of strange to give a talk like this. I'm still not used to giving talks online, but I will do my best. Okay, so the title of my talk is Overview of the HDAC6 Discovery Program at Horizon. Okay, just a quick legal notice. We're supposed to do this because we're a publicly traded company. With a disclaimer, I'm supposed to show this. Okay, so Horizon Genomics is based in Barcelona. For those of you who don't know anything about us or where we're placed, Barcelona is in the northern part of Spain. We're next to the French border, and it's one of the most active biotech hubs in Spain. Some of the, I would say, biggest research infrastructures in drug discovery that can be applied to drug discovery are based, nearby. I'm just showing the Barcelona Supercomputing Center, where one of the biggest supercomputers in Europe is located, and then we've got this particle accelerator as well, quite close by. And we've got plenty of science, big science parks. So we're in the middle of this, soup, so to speak, which is always good for a biotech company. Okay, so what we do is we develop small molecule therapeutics. Just the talk that was given by Antonio Gomez, our friend Antonio Gomez from Janssen, was quite good. To give some context, we're right in the middle of this approach. So we develop small molecule therapeutics, especially in the epigenetics area. And we are very focused into only two different therapeutic areas. One of them is oncology, and the second one is CNS. We're a clinical stage company. We've got two clinical stage assets. Both of them are in phase 2. I will talk about this later on. And we've got a growing group of early discovery projects, and I will be talking about one of them. These are just a few figures of our company. So as I said, we're publicly traded. So our pipeline is the one you can see on screen. So as I said, we've got two compounds in phase 2. Both of them are targeted to LSD1, which is this stands for lysine-specific demethylase 1. It's an epigenetics target. And so we've got ORY-1001, which is called also Yedademstat. This is currently being tested in acute myeloid leukemia and small cell lung cancer. And we've got a second one, which was optimized for CNS, targeted to CNS, which is currently being tested in many different CNS conditions. We just started this Portico clinical trial in borderline personality disorder. Okay, so other than this, as I said, we've got many other, early stage programs, and I will be talking about this one, which is revolves around the design of HDAC6 inhibitors. Okay, just to give again some context, as I said, we're working in the epigenetics field For those of you who don't know much about this means, epigenetics has been defined as the study of heritable phenotype changes that do not involve alterations in DNA sequence. Basically, what this means is epigenetics is kind of an umbrella term that engulfs, all the processes that involve, in a way, covalent modification of DNA, especially via methylation of cytosines, or covalent modification and on the histones, which are involved in packing and repacking DNA, and this has a direct impact in gene expression. Basically, when we talk about epigenetics targets, you've got a series of different families. The most interesting, I would say, traditional ones are writers, erasers, and readers. So writers are enzymes that somehow add covalent modifications on DNA or histones. In this group, you've got DNA methyltransferases, lysine methyltransferases, and histone acetyltransferases. And then you've got a second group of enzymes which take those covalent modifications and remove them. In this case, you've got, for example, the lysine demethylases and the histone diacetylases. I will be talking about a member of this family. And then, other than adding or removing, you've got a series of proteins that are able to read, to interact with these histones when these histones are somehow tagged with these covalent modifications. Okay, so these are the three main families. So I would say epigenetics is becoming more and more mature, but still, I would say it's a bit unexplored territory yet. There are lots of different mechanisms that are not known, which play a big role in the development of a series of diseases. Definitely, many conditions that fall within, our two main therapeutic areas, oncology and CNS. And when you have a look at, what has been approved by the FDA, any molecules that somehow modulate writers, erasers, or readers, you've got this group here. And if you have a look, the most populated group is, indeed the one that modulates histone deacetylases. So vorinostat, belinostat, panobinostat, and romidepsin have been approved for different indications. These are their structures on the right-hand side. And if you notice, most of them, 3 out of 4, do indeed have hydroxamic acids, which have, certain tox liabilities, which I would say are not, they are drug-like, but not so much. So they're not highly desirable, so to speak. So when we move to this family of erasers, so the histone deacetylases, this is the enzymatic activity they carry out. So basically, you've got a tagged lysine with this acetyl group, and what they do is they remove this acetyl group, and you go back to an unmodified lysine in terms of the side chain. And you've got basically 2 big families: the zinc-dependent family of histone deacetylases and the NAD+ dependent family. And HDAC6, which is the one I will be talking about today, falls in this class 2B, so it's a zinc-dependent. So in principle, there are many other members of this family which are quite close in terms of sequence and structure, but there are differences that allow to design highly, or at least this was the approach at first, to design highly active and highly selective compounds. Still, I would say HDAC6 is kind of an outlier in its own right, because it's not located in the nucleus as many others, and does not deacetylate specifically, the histones that are involved in packing DNA, but it's got instead a series of substrates which are located mainly in the cytoplasm. I will be talking about this later. So, as I said, it's kind of an outlier in this family. Basically, it's involved in many physiological processes. I'm talking about HDAC6 now. Basically, I would say the 2 most important ones are intracellular transport And the second one is protein homeostasis, so elimination of protein aggregates. One key thing that we considered when we started this project is that, and this is good, it's a good feature of this target, is that the knockout mice are viable. So you knock out HDAC6 and you still have a perfectly viable animal. And this is giving you a hint on maybe lower uncertainty in terms of on-target tox effects. And the inhibition of this enzyme has been linked, if you have a look at literature, it's been linked to all sorts of different, as a promising therapeutic approach to all sorts of different indications, from cancer, ranging from cancer to cystic fibrosis, neuropathy, peripheral neuropathies, and the like. Okay, so with these approach in mind, we wanted to design very much, as Antonio Gomez was saying, we wanted to design small molecules that could modulate this enzyme, that could have high potency and also high selectivity, and we wanted to keep away from the hydroxamic acid. So we concentrated our efforts in 1,2,4 oxadiazoles. Okay, so this funnel that you usually see in the presentations, we obviously applied. This is just the screening cascade that we applied that allow you to go from these thousands and thousands of molecules to just a few. Basically, as a first step, we checked for HDAC6 biochemical activity. Obviously, you want to have a highly potent compound. Then you move on to cellular selectivity and cytotoxicity. You want to make sure that the compound is not cytotoxic. Then we tested cellular EC50. We very early on checked for hERG inhibition. Obviously, this is done routinely now in all drug discovery projects. Then we checked for microsomal stability and P450 inhibition. You don't want to mess with the cytochromes. And also we were checking very early on activity in other HDAC members other than HDAC6. We wanted to hit specifically HDAC6. And then, once this was done, we moved to pharmacokinetics and aims in micronuclei tests. You want to make sure that your compound looks good in vitro in terms of tox, and then maximum tolerated dose, and also a diversity profile to make sure that the compound, other than other HDACs, is not hitting the usual families of receptors such as GPCRs, kinases, and the like. Okay, so this is the bird's view of the whole of the program. We ended up, it's like a retrospect view, obviously. We ended up synthesizing around 350 compounds, all of them, around this 1,2,4 oxadiazole core. And by applying all sorts of filters, the ones that I just showed, we ended up selecting only 2 pre-candidates. So these are the 2 molecules that, in our, from our perspective, were the best among all those synthesized, and had, let's say, promising results in all this screening cascade. And I will be showing you over the next few slides, their full characterization. So right now we've got these 2 clinical, preclinical selected compounds. Okay, so we start with, well, by the way, their code numbers are OG4652 and OG4804. So we checked, obviously, the first step was checking activity on target for HDAC6. Both of them are highly potent. They're around 30 nanomolar. By the way, both of them belong to the same chemical series. So, both of them do have the same chemical core. The difference between them is that they've got different, decorations, outside decorations, but for the most part, it's the same chemical core. Once we checked that these 2 compounds were very highly active on target, we wanted to make sure that the compounds are indeed selective for towards other HDACs. And this is the case. As you can see, neither of the compounds seem to be hitting the other histone diacetylases. And by the way, we were always checking. This is also routinely done in a drug discovery project, we were checking how we were comparing to this research tool, to Tubastatin A, which has been described to be highly active and selective, although it does indeed feature hydroxamic acid. And the second compound is ACY1215. This is a compound that's from a company called Acetylone, and it's been described as a highly active and highly selective compound. In our hands, this was not so. So when we compared our two compounds to this Acetylone compound, our compound seemed to be more selective. So in terms of biochemical activity, they looked good. So we moved also to the panel of varied targets. We tested this in the Eurofins CEDEP. So we tested both of our compounds in a panel of 100 different targets. You can imagine this is composed of GPCRs, nuclear hormone receptors, kinases, and the like. So for OG4652, seemed to be hitting only 2 out of these 90 plus nearly 100 targets at 10 micromolar. These are the two receptors that seem to be hitting, but only around 50% inhibition at 10 micromolar. So it's not that bad. And this second compound, our second pre-candidate, 4804, only hit 1 out of these 94 targets. So yeah, this is pretty reasonable. This is telling you that our compound is not only highly active on HDAC6 and highly selective towards other HDACs, but it's also not very promiscuous. It's not hitting many other kinds of receptors. We also run a very small panel on epigenetic targets, and 4804 seemed to be hitting no targets, and 4652 only hit these lysine demethylase. So again, this seems reasonable. Okay, so we checked in cellular assays. We wanted to check that our compound is indeed inhibiting the deacetylation of tubulin, which is one of the main substrates of HDAC6, but it's not hitting the deacetylation of, for example, histones, H3 and H4, which are deacetylated by other HDACs. Okay, and this is a crowded slide, but just to make a long story short, we applied a potency selectivity ratio for both of our compounds, and we compared again with this acetylone compound and tubastatin, and our compounds seem to be to have a let's say more promising profile in terms of potency and selectivity. Okay, so we move to cellular dose response. Again, here what we want to see is that our compounds do indeed have activity in terms of inhibiting deacetylation of alpha tubulin, and this was the case. These are the EC50s for both of the pre-candidates. Both of them are submicromolar. One of them is 0.3 macromolar, and the second one is 0.1 micromolar. And then we did the same, but with histone H3, which we don't want to mess with. And the result was quite good, because neither of the compounds seemed to be, active in these cell-based assays. So basically, this is telling us that our compounds are indeed inhibiting the deacetylation of tubulin, but are not inhibiting the deacetylation of histone. Okay, so I'm just showing this as an example of the progress that you see in a drug discovery setting, right? you keep on applying more and more filters. So as we applied all these filters, many of the compounds that we had synthesized, were left aside because of unwanted or not promising data. Okay, so in terms of in vitro admit, we wanted to check, obviously, microsomal stability in human, mouse, rat, and dog. We checked for intrinsic clearance. You're also supposed to be checking for plasma protein binding, and also this CYP inhibition. So you don't want to mess with the cytochromes. And also this is the CACO2, so the permeability data for these two compounds. So basically, as you can see, both of the compounds are quite permeable. One of them is it does indeed have, let's say, moderate permeability. The second one is very permeable. And none of them were messing with a P450, so they have no percentage inhibition at 10 micromolar, so this is good. And the plasma protein binding was, I'd say, fairly reasonable, not too high and not too low, which is also, again, what we were looking for. So this was, these are nice results. Moving on other tests for in vitro ADMET, we checked for cytotoxicity. Both of them seem to be, okay, no cytotoxicity. We were checking for hERG. For one of the compounds, for 4652, hERG was around 50% inhibition at 30 micromolar, which is already quite high. So we could say this is fairly reasonable, although, it might be desirable to have a lower value. For the second one was very low, so this is good. And then in terms of mutagenicity, you're supposed to, apply these Ames and micronuclei tests, make sure that your compounds are not mutagenic, and this was indeed the case. So both of the compounds look very reasonable in terms of ADMET. Once you've checked for this, so you know that the compounds are active, they're quite selective, they do have, reasonable solubility, permeability, and so on, you move on to pharmacokinetic studies. This is usually done in rodent, mice, or rat. And then once you've checked this, you're supposed to, move on to a bigger species such as dog and so on. I will be only showing you the data that we've got for mice for both of the compounds, again, 4652 and 4804. Sorry, both of the compounds seem to have moderate plasma clearance, and both of them do indeed have, let's say, fairly high bioavailability, meaning, this is good, okay? So your compounds do indeed have, let's say, desirable pharmacokinetics. So they have good oral bioavailability, and again, they were considered to be, in a stage where, they can be progressed forwards. Okay, in terms of MTD studies, we also obviously wanted to check exposure. When we do this, usually we take exposure in different tissues, once they've been administered in mice. And usually what you do is you take at least one peripheral tissue and then brain. And these are, the preliminary data that we got for both of the compounds in different colors at 2 different doses. So this is 10 mg per kg and this is 30 mg per kg. And as you can see in lung, peripheral tissue, both of them do have a nice exposure. But when we moved to brain, one of them, which was 4652, seemed to have higher exposure in brain. Meaning, if we're to believe this data, we're doing further experiments now. But if you're to believe this data, this is telling you that one of our precandidates does indeed reach the brain better than the other one. Okay, so obviously, whenever you're designing new compounds targeted at a certain macromolecule, in this case HDAC6, you want to make sure that you do have target engagement, both in cell-based assays and in vivo. In this case, I'm just showing you the MTD data that we got for target engagement for both compounds. Again, this is 4652 and 4804. So as you can see, this is like an indirect way of checking for target engagement. We were checking acetylation of alpha tubulin, which is, as I said, substrate of HDAC6. And in moving from, when you move to higher and higher concentrations, this is 10 mg per kg, 30 and 90 mg per kg, you do indeed have, a very nice dose response in terms of, acetylation of tubulin. And this is not the case when you move to checking acetylation of histone H3, which is, again, not its primary substrate. So this was, again, nice result. And finally, we preliminary data in 5-day administration in mice. This was only done for 4652, not 4804, but we wanted to check the tox effects of the hematological and liver levels. So this is also routine. This RBC stands for red blood cells. This is platelets, PLT, and these are neutrophils. And these are 3 aminotransferases that are typically checked. Also ALT, AST, and GGT. And these are the different dosing regimes. So this is 10 mg per kg, 30 mg per kg, and 90 mg per kg, which is, let's say a fairly high dose. And even in the high doses, you can see that the counts for the red blood cells, platelets, neutrophils were pretty much the same. This is telling you that you go to higher and higher doses, and you still have no toxicological effect in the level of blood. And then we did the same for the aminotransferases, and again, on moving to higher and higher doses, the results seem to be quite reassuring in terms of not messing around with the transferases. So to conclude, we do indeed have 2 novel, highly active and selective HDAC6 inhibitors. We've designed them, and we've checked that they've got high activity on target. They have a high selectivity towards other HDACs. They have very reasonable ADMET profiles. They also have good pharmacokinetics in mice with high bioavailability. The preliminary data in maximum tolerated doses at 5 days in mouse are also good. And if we were to, highlight any difference between these 2, I would say that one of them seems to be more prone to reaching the brain than the other. And this is just as a summary. We've checked, the activity in clinical stage. At the clinical stage, some of the compounds do indeed target HDAC6, and these are the ones that we have found in the clinics. So this company, Kairos Therapeutics, does indeed have a compound in the clinics targeted to HDAC6. This is for cancer. And then the most active company is Chongkundang, the Korean company, which has 3 different compounds in clinical stages. One of them is for Huntington. The second one is for rheumatoid arthritis. And the third one is for Charcot-Marie tooth, which is a peripheral neuropathy. And with this, I will conclude my talk, and I'm happy to answer any questions.
Hello, Robert. Thank you so much for your insightful presentation. I do have a question for you. So, Robert, can you tell me what are the new strategies Oryzon Genomics following for screening and discovery?
Well, so, we have a series of ideas that we're exploring. Always in the epigenetics field, and as Antonio was explaining during his presentation, we do indeed use lots of different technologies, from in silico technologies, because I have a very strong background in computational chemistry applied to drug discovery. We do have certain alliances with a series of companies. That's why, for example, I found the previous talk very interesting, definitely. But in terms of epigenetics, I mean, there's in science, I must say, this is personal view. I might be wrong, but many people also move in waves, and it seems like people consider epigenetics was like a past wave, and we are not aligned with that, because we know, because we're fully focused in epigenetics, and we know that there's still so much to be learned from from epigenetics, and we're very keen on exploiting this.
Okay, got it. So, Robert, thank you so much for your insightful presentation. We are very happy to have you as a keynote speaker, and looking forward to work with you again. And okay. I will ask the audience if they have any question, they can ask you on the main stage chat. Thank you so much, Robert. Okay, so ladies and gentlemen, our next presenter for today's presentation is Dan D. Aridor. He will be with us in a moment. Stay connected. Morning, Dan. How are you?
Good morning. How are you?
I'm good. How are you?
I'm good. Thank you.
That's great.
Excited to be here.
Thank you so much, Dan, for joining us. We are honored to have you as our keynote speaker. So I can see your slides. I can see your screen. It's perfectly all right.
Can you see it?
Can you click on your slides? Can you go to your slides? Yes, I can see it now. It's perfectly all right. So, ladies and gentlemen, now with us is Dan D. Aridor. He is the chairman of Aebi-Bio from Israel, and he's here. So, Dan, you can start your presentation now. Stage is all yours.
Okay, thank you. Thank you for coming to hear me. I'm really honored. If I would have told you a year ago that within 1 year, the world will find a vaccine for COVID, which we have not heard before, you would probably call me crazy. I think we are seeing an amazing development in science and technology. And I want to talk to you about why plans are useless, but planning is indispensable, and my experience and how I see it. Development of drug discovery and development at all is usually not a linear projection, and many times discovering things is an accident or a way of moving forward. We've seen it in drugs many times, and I think the story of mRNA is a great example of how once a neglected technology is being used today, basically to save the world. The problem is that usually when you discover something so fundamental, most of the time it's unappreciated. And we have many examples, including the story of mRNA. This is a work of 3 decades. And at that 3 decades, her work has been belittled, neglected. She even lost her position in the academia. And the story was that she had a great idea and a great technology. She worked for 2 decades. The problem always, and not always, but sometimes with drug discovery, is that you find a solution and then you might have a problem of delivery. And this is usually what you can see in many drugs. But you worked on the basic idea, and then also the way to deliver the drug and to finish the product was availing itself. So already in 1995, after years of working, she was belittled and actually dismissed from her work in academia, and said to her that there's nothing there, there's no gravitation, there's no pull for this. She's working on something which is basically useless. But she did get noticed, and the idea was that after she developed the primary concept, the idea how to deliver it and solve all the other problems was also availing itself. And last year was a great example how everything just clicked together to becoming the world vaccination and huge technology breakthrough. And this is an effort of not of her, but also the entire academia and the entire scientific community. And this is what's great about it. But without her and without her great effort and a great tenacity and a great insistence on what she was doing, we wouldn't be in this position today. And I think she's a great hero and a great insight to and a great honor to what she has done is absolutely amazing. His story will be told for many generations to come. This is not the only story of such drug. I think that if you can recall the Merck blockbuster, which is being sold today about $10 billion, is also such a story. When Merck bought the company that had the PD-L1 inhibitor, they did not buy it for the PD-L1 inhibitor. They bought it for other reasons. They did some sort of internal review and gave this project a value of 0. Only when they heard that another company is doing something similar did they start to, they changed the concept and gave it the money and the timing in order to make it a blockbuster. But it was basically really accidental. The other company, if I recall, GSK, basically failed with the experiment, and Merck has $10 billion, the best drug today for cancer. And I think even the originators of the idea never thought that this would be the story of what they have found. They were actually looking for something totally different. One of the great examples is actually the phage display. Phage display was supposed to be the biggest drug discovery platform, and 30 years ago, this was supposed to be it. This was supposed to solve all the problems. This was supposed to give us the ability to work with antibodies in a very specific way. And the phage display inventor actually got a Nobel Prize in chemistry, but it never fulfilled the dream. And the question is why? Why something which was planned to be so good never really happened? And this is more than once a real big story in science. It never rarely comes out in a straight line. And this is also the beauty of it. So when Dr. Ilan Murad started after his post-PhD in Harvard Medical School, he started in a company that used the phage display technology, and it's called Peptor, and he was working on the phage display for 3 years and basically coming up with nothing. So he went into the Weizmann incubator with an idea how to fix the phage display and how to make it work. And the phage display is a great technology, but it wasn't really working. And the question was why. And what Dr. Murad and Dr. Itzhaki were able to do were three things. First, they were able to eliminate the deletion mutants in each round. Second, they were able to split the round into one affinity and one efficacy. And this made a great difference, because once you don't have a limitation of the number of rounds and you also target efficacy and affinity, you can end up with a very specific peptide that you work. And what they have changed is also they are not using solid support, and instead of using antibodies, they are using peptides. And this is huge because antibodies are relatively big and they are really strong, but they have their own will and sometimes hard to manufacture and can connect in points where you don't want to. So if you can have a peptide which is 10 or 12 amino acids long and does exactly what you want, then you have a huge advantage. The problem is, how do you make sure that it has strong enough efficacy like a antibody? And that's the next stage of what Dr. Murad and Dr. Itzhaki did. It took about 10 years to develop this platform into perfection, and we are still perfecting it. And when we started, the peptides were not the big stuff. They were considered to have many issues and many problems. One of them, of course, is the delivery, exactly like the mRNA. So I'm just going to give you an overview of what happened. It took us about 2 decades. And this, I think, what happens with great discoveries. It takes time. And if you don't give it enough time, it will not happen. This is also the story why big pharma rarely innovate. Because what happens is that if you start a project in big pharma and you need to give an answer and you get to give a delivery in 4 or 5 years, and then the CEO changes and the project ditched, and then it goes out of the window. So what happens, big pharma rarely really innovate from within, but by innovation outside. This is also the story of mRNA, of course, and also of Merck, PDL1 and others. This is what happened when we took one peptide and tried it in mice. Basically, we had a few minutes of efficacy. And then the idea came how to prolong the idea. And this is what Dr. Murad told me in 2006, when I just joined ABI, is that eventually down the line, we will find a way to prolong the efficacy of the peptides. And this is what he did. He took basically a peg and put on the peg peptides and prolonged it from a few minutes to a few hours. And this solved the problem of delivery. But it's not it did not solve everything. So what happened next is that we took this peptide with target EGFR targeting peptides into mice with lung cancer humanized version. And this is what happened. Basically, you cleared it out of the kidney, you cleared it out of the liver, and then it stayed in the tumor. And this is just one shot. So you see the accumulation has stayed flat because it was just shot. It just stayed on target. This is how you see it. You're using the PEG. Basically, you are able to zoom in on the cancer, and after 24 hours, you see all the PEG, all the EGFR targeting peptide inside the cells. So this solves the issue of delivery. But how do you make sure that you only target the cancer? So here comes the great advantage of the academia and the fact that everybody's building up on other people's work. Dr. Pavel Kitov and Dr. Dave Bundle from the University of Alberta found the avidity effect. And the avidity effect says basically that if you are able to put two different targeting compounds on a different scaffold, and they use antibodies, you will be able to get an effect which says that the number of interactions between the cancer target and the compound is much higher than the number of receptors. And we have to understand that the number of receptors in cancer is many times more than the normal size. For example, if you take EGFR, then EGFR is 100K number of receptors, and normal cells is just on singular numbers. So you have this is solving one problem. But then what Dr. Murad and Dr. Itzhaki did is basically targeted 2 and 3 receptors at the same time, and this gave it the full avidity effect of what they wanted to do, because once you target 2 or 3 receptors at the same time, the number of interaction is exponential, and this gave us the ability to target only cancer cells. But the problem was still how to use it very safely, and how to not just stop the interaction, but also stop the cancer itself, because cancer mutate. The cancer cannot mutate as far as we know. It can mutate once, it can mutate twice, but as far as we know, it cannot mutate 3 times at the same time in terms of dropping up the three receptors at the same time. But yet we wanted to get a much more strong effect. So at the beginning, we started by using a natural toxin, but the natural toxin was a big antibody or part of it, and then it was able to penetrate the cells itself. So what we did was using the platform to discover new peptides on the same target. So we added to this compound a peptide toxin, which was 10 or 12 amino acids long, and also it only worked inside the cell. So you have a binary concept in which the targeted peptides are actually discussing themselves, are actually interacting only with cancer, and then they internalize the entire structure, and then the toxin peptide works. And this gave us the desired effect without the side effects that we wanted. And what you can see is that in a different experiments that we did, we changed the number of targeting peptides, and then we got a much higher effect with much lower dosage. As a control, we did some sort of instead of 100K EGFR receptors, only 3,000, to see the fact that the effect is only working via the process that we wanted. And this is a PBS vehicle control effect that you see on 3,000 targeted receptors. The effect is much lower. And this gave us the understanding that this is what is working. One of the hardest targets that you have is the cancer with a RAS mutation. And when ABI and Dr. Murad and Dr. Itzhaki tested their mutato concept, they actually tested it on the humanized RAS mutation, and this was huge because not only did we test it on the RAS mutation, we did it on a solid tumor. So instead of trying to stop any sort of signal transaction, the fact that we were able to internalize the entire structure and then release the toxin to do its work basically solved the entire problem of the ripple effect of the RAS mutation by totally bypassing the whole problem. What we are seeing now is that other people, as Dr. Morad foresaw maybe more than a decade ago, are starting to look into the receptors and the receptors as an answer, because this is the one common denominator of all cancer, that you have a lot of receptors, different receptors, but a lot of receptors. And the way to internalize is via the receptors. Also with immune therapy. And we are seeing more and more now people coming through with this concept of having to go with the receptors and also trying to attack 2 or 3 times at the same time in order to get this effect. I'm very happy about this because when I joined the company in 2006 and 2007, when this whole concept was just beginning to formulate, people were belittling peptides and belittling receptors. And we didn't have the exact concept at that time. We just knew that we have a great technology and we have a very basic concept of how to play with amino acids. And we thought it was worth a lot. And we thought that a solution will avail themselves. The problem is that we never thought it will go through this via dolorosa that we had to. We thought that once we prove the efficacy of the platform, we will be able to share the fruit of this concept with the entire community. And what we found out, that even though we proved the concept on various levels, every time we wanted to collaborate and every time we wanted to work with either big pharma or funds or whatever, every time they just raised the bar and said, okay, now you do with different target. Now you do it with mice. Now you do it with humans. And this is basically where we are going. We showed in mice, and I'll show it to you in a minute. So by the way, all experiments were done outside of ABI in vivo, everything you see. So we took the peg and put EGFR targeting, PDL targeting, and c-Met targeting, and we injected to the tail vein on a solid tumor. But instead of just using also chemotherapy and other agent, we just did it for 3 weeks. And these are results. We show a big difference in the progression of the tumor. Using only this, and we also checked back the peg, and it was, when we checked it back, it was only 60%, had only 60% of number of arms which were working. So we can repeat it, but this time with a better peg, and then COVID crashed, and we were cut off from supply lines, and that was horrible. So here you have plans and nothing really happens. Just for example, if you took other companies' experiments, they're not doing better on their own, only when they are using much higher dosage and other compounds. And this gives us a lot of hope that if we would have used just chemotherapy as a add-on, or just increase the dosage, we will be doing even better without the side effects. So what mutato really does is that it stops the cancer on its track immediately. We also believe it will also stop the mutations, because we are going to attack also the possibility of the mother cells, because the difference between them and regular cancer is only one mutation. We tested it, and none of the mice that we injected it with had any side effects. Other companies had basically mice dying on these experiments. Yes, it's only mice, and everybody has cured cancer in mice many times, but this is something we have to show in order to move forward. and we tested on solid tumors. The basic idea is that in order to solve cancer, you have about 100 receptors which are shared. And if you will have 100 targeting peptides which are the same as a library, you can always play and click with it and do it both as a generic and as a personalized, because many of the cancers will be repeated. For example, we started with the PD-L1, EGFR, and c-Met, which are very common in many cancers. So we can have on a shelf 10 or 20 already approved mutators, and then just play with the receptors according to specifics of a person. We are now trying to move forward with the first mutator, which is EGFR, PD-L1, and c-Met, which applies to head and neck cancers, lung cancer, and colon cancer. And the results that you've seen before are for that. Here are where plans are useless. We believe that once we will publish the concept, we will be able to raise the funds. And what happened, we published in February 20th, and we had already discussions in March with people from England and venture funds, and then England just shut down and we were devastated. Then we started to raise money in California, and California was shut down. And now we're trying back after a bit of time has passed and people are starting to relax to raise funds again. And you can't imagine how much a break heart it is to work on such a long time, and then you have something so extraordinary and unpredictable coming and just crushing everything you worked for. And that was devastating what happened with COVID. And it's kind of heartbreaking, because if we are right and this could be a huge help into cancer patients, then the lockdowns and the total stop of the economy has basically delayed a solution which can save many, many people. And as somebody who's worked on this for 15 years, this is heartbreaking. At the beginning of the year, we saw again that people are starting to, I don't know if it's related to us or not, but to look into the concept of double and triple attack at the same time, and getting the results that we predicted will be happening, but they're not putting it on the same structure. And the reason they're not putting it on the same structure, because they're using mostly artificial, natural peptides, which are longer, and we are using artificially designed peptides, which are smaller, and we are able to put them on the same scaffold without them being interacting with each other or in the way of any sort of way, and they are very, very precise. So we are sort of waiting for the whole world to catch up with what we are saying without being rejected and belittled, and this is taking time, unfortunately. During 2019, we were already in discussions with Charles River just to finish our preclinical tests, and those preclinical tests should have taken about 10 months. Now they're saying 8 months, in which they just want to show that there is no side effects or very little, and no immune response. And we already started some initial tests in Israel, and we are seeing that both assumptions are right. The concept is very clean. The concept does not create immunotherapy or negative side effects. And what we just need, and this is what killed us, is that does $5 million to finish this work. And I'm really shocked at the fact that, despite the fact that we are not coming from academia, despite the fact that we are worked on this outside the normal course of events, this is what's happening, and we are not able to get the $5 million just to prove that this concept works. I hope this presentation will help me get the money. We are a private company. We funded the company for almost 15 years on our own, with very low salaries, working on a great idea. And the fact that we are not coming from, Dr. Murad and Dr. Ishaki are coming from academia, but worked in ABI for 15 years and not published much, that is basically is a source of ridicule and belittling, and it's not. It happened before in the drug discovery, and I'm really devastated and disappointed with that. Sarina, I have enough time? Hello?
Yes, yeah. Dan, you still have 5 minutes.
Okay. I think that this is one of the things that I'm finding difficult, is that discovery is sometimes a breakthrough, but then the whole community has to catch up. And I'm not the inventor, so I can speak about it without being arrogant. I think that what Dr. Murad and Dr. Itzhaki did was an amazing breakthrough, and I think they were not given the appropriate respect and the appropriate attention to what they have done. And they have proved the concept in mice. They have proved the concept outside of ABI lab. And the fact that they have been ridiculed or belittled is, in my opinion, actually costing life. I want to tell you just a small story of Maria Copecci, who was awarded the Nobel for a concept he was ridiculed for. When he finished Harvard Medical School, he applied for 3 grants from NIH, and the NIH told him, and they knew who he was already considered a genius. 2 of the concepts are really good, the third one is disastrous, but since you put it in such unequivalent terms, we will give you the money for all 3. Maria Copecci took all the money and put it in the third one, which was considered a lost cause, and he won a Nobel on that. When he was interviewed later on, he said, and he refused. By the way, he refused to continue his work in Harvard. He started his work in Mormon land. He said he thought that if you really want to do something great, you have to give it a long time. And this is exactly the story of mRNA. This is exactly the story of the PDL1. This is exactly the story of Maria Copecci. And you can't deliver something really big at a short time. And when people come to us, well, it took you a long time to develop this. So I tell him, what do you want us to do? It took a long time. Yes, it did take a long time, but we have the results. Why does it matter? I hope that the fact that the world has seen in a year the development of a cure for COVID or a vaccine for cure of COVID will change the concept about the ability to do something huge also in cancer, because the science here is here, the proof is here, and we published it, and the money that we need is not that huge. And I hope this will be a good sign to start the new rounds of funding. I think that what summarizes it, that we choose to go to the moon in the decade and do other things, not because they are easy, but because they are hard. And I think for the last 15 years, we choose to do a really hard work, and we show the results for it. And I hope that the new environment in which bio is a big thing can give us the boost that we need. I thank you for your attention, and if you have any questions. Sarina?
Yes, Dan. Actually, thank you so much for your presentation, and I have a question for you. Sure. So, approximately when we can expect this vaccine in the market?
Well, if we have the 5 million from the moment we have it, it will take us about a year to be able to be in humans. That's about a year and a half to prove the concept in humans.
Okay, that's great. So Dan, it's actually a very good concept, and it's a very good thing that you are taking step on this. So I would ask anybody in this connection if they want to invest with you.
I'm sorry?
Okay, so Dan, if somebody will have question, they will be asking you on the main stage. And thank you so much for your presentation. It was quite knowledgeable session. Thank you so much. We are looking forward to work with you again. Good luck.
Thank you so much.
Thank you. Bye. Okay, so ladies and gentlemen, our next presenter is Eva Mendez, who is drug discovery head at Oncoheroes Biosciences, and she will be with us in a moment. So stay connected. Thank you so much. Okay, so ladies and gentlemen, there are some issues Eva is facing. So she's just coming in a moment, so stay connected. So I would like to ask you that the topic which has Dan explained with us about the mutato. So I think it's a very good step, and I hope that some of you would be thinking about to invest with him for the mutato completion.
Yes?
Okay, yes,
Okay.
yes, perfect.
Sorry for that. Okay. Okay, so I need to share. Yes?
Yes.
Okay.
Okay, hello, Eva. How are you?
Fine. Sorry for this. For the internet issue that I have. I'm really sorry about that.
No, it's okay. It's all right. So thank you so much, Eva, for joining us. It's an honor for us to have you as a keynote speaker. So ladies and gentlemen, let's welcome Eva Mendez. She's working as a head of drug discovery at Oncoheroes Biosciences. So Eva, you can start your presentation. The stage is all yours.
Okay, so thank you very much, and again, apologize for the delay because I had some internet issues, and thank you for inviting me to just present the work that we are doing in our drug discovery division in Barcelona.
Right.
So first of all, let me introduce the company. So we are a biotech company, 100% focused in discovering, developing, and commercializing drugs for children and adolescents with cancer. So the company was incorporated in August 2017 in Boston, and in March 2018, Oncoheroes Europe was incorporated in Barcelona, Spain. So we have, as you see here, we have two divisions. We have the drug discovery division that is based in Barcelona, and we have the clinical division that is located in Boston. So the clinical division is focused on in-licensing clinical or almost clinical ready assets to develop them. And in the drug discovery division, we are focused on discovering new oncologic targets and creating our own IP. So we start our first project in summer 2018 with the funding from the National Foundation for Cancer Research. So in that summer, in summer 2018, we opened our lab in Barcelona and we hire our first scientist. And we start our first project, this one that I'm going to present. So this project is focused on medulloblastoma. And why medulloblastoma? Because it's the most common brain tumor in children, accounting for almost 20% of all the CNS tumors in children. It's also responsible, as you see here, for almost 10% of all pediatric cancer deaths. And the current treatment is multimodal, so it includes surgery, it includes also radiation and chemotherapy. And despite the treatment improvements, 40% of the patients do not survive. And those that survive, they will have severe secondary effects because of the treatment. So our aim is to develop new treatments for medulloblastoma with greater therapeutic effect and lower toxicity. Okay, so medulloblastoma. Okay, so it's divided into 4 different molecular subgroups, with different biomarkers, and mainly different outcomes. So we have here the winless and the Sony Hedgehog groups, and we have group 3 and group 4. We have focused on group 3 that accounts for 25 or 30% of the cases, and it's because it's the more deadliest of all the molecular subgroups. So it accounts for especially if it's associated with MYC amplification, where we only have 20% or survival 5 years post-diagnosis. As you see here on the right hand side, it's found in infants and children, but we don't see it in adults. So we have focused on this molecular subgroup, and we will develop new targeted drugs that will benefit this specific group 3. Okay, so our therapeutic approach is the synthetic lethality that refers to the death of cells caused by concomitant perturbation of two genes, each of which is non-lethal alone. And our strategy includes the pharmacological blockage of this driver gene and also the target gene, or the combination of a mutation and a pharmacological blockage of this target gene. So with this approach, we expect to target two targets simultaneously, and we hope to increase the therapeutic efficacy and reduce toxicity and resistance. So which is our strategy? Is to develop a drug discovery project with an initial goal of selecting and characterizing a preclinical candidate to be ready to enter the regulatory preclinical phase in 2023. This preclinical candidate will be a dual compounds that will target both targets, or a combination of compounds that exhibit in vitro activity against the two targets and in vivo as well as a suitable pharmacokinetic profile and no concerns of preliminary toxicity. Of course, our ultimate goal is to develop new treatments specifically designed for these medulloblastoma group 3 children. And as we are really aware that the ADME profile is really important and we can reduce the high attrition rate in the clinics, we include very early in our screening cascade, we include extensive ADME tox profiling assays in order to prioritize and advance the hits, not only based on activity, but also on ADME tox properties, okay? So here are the models that we are working with. So on the bottom, you can see that we have developed a screening platform. It's a 2D. It's developed in microtiter plate, 384 well plate format, and also with the standards of the pharma industry. So we are taking really seriously the sensitivity and robustness of the assays. And we have included a panel of 8 medulloblastoma group 3 cell lines in order to increase the efficacy of the potential treatment. And we have also included 3 control cell lines from tumor-specific environment to avoid toxicity. As you will see later on, we are really, really aware of the toxicity, and we include as soon as we test for efficacy and toxicity all the time from the very beginning. So this is our 2D screening platform. Some of the medulloblastoma group cell lines we have acquired through an agreement with Duke University. And then we have included also several in vitro toxicity models. So we have a general toxicity model based on BG's line, and we have also developed 3D neurotoxicity models. So we have developed models for neurons and glia cells in 3D, and we have also included pathotoxicity model just to be sure that we don't have any concerns. We have in mind to set up also a cardiotox model in the following, let's say, weeks. And regarding the in vitro efficacy models, as I mentioned before, we have set up the 3D monocultures conditions for the medulloblastoma cell lines and also the control cell lines. And we are just finishing the conditions for the co-cultures of the medulloblastoma and control cell lines. We are also developing cell lines resistant to the standard of care. We have started with the cisplatin, but we will include different resistant cell lines. And we also, as we are treating brain tumors, we are going to include also a BBB model. But we are not only thinking on a physiological BBB model, but in a physiopathological BBB model. So we will include not only the endothelial and neural cells, but we will also include the medulloblastoma cells. So we will see if the compound is able to penetrate the BBB, but also to have efficacy in our medulloblastoma cells. So as soon as we finish the development of the co-cultures in 3D, we will start with the microfluidics just to have a more translational models and just to control the oxygen and the nutrients fluid. And For regarding the in vivo efficacy models, so we have developed the subcutaneous one and we have validated it with cisplatin, and we are just about to finish our validation of the orthotopic model with intracerebellum implantation of medulloblastoma cells. And we are just about to validate it with cisplatin. So as you see, we have developed highly a novel platform that really integrates a broad panel of in vitro and in vivo human models, specifically designed for medulloblastoma group 3. Okay, so here, as I mentioned, and I will be talking about this through the presentation, we are treating a brain tumor. So we are really aware of the potential neurotoxicity that we can observe. So what we have done is start with the Lumes cell line that we have successfully differentiated into neurons, as you can see here. But we have also included neural stem cells. Why? Because it's the putative origin for the medulloblastoma group 3 cells, and also because we have successfully managed to differentiate them not only into neurons as the Lume cells, but also in the glia cells. So we have the astrocytes and oligocytes, dendrocytes. So here you have the spontaneous differentiation those are in-house images, and here you have the directed differentiation where you can observe both type of glia cells. Okay, so regarding the target ID, we have considered two different approaches. One is the experimental approach, so the in vitro approach, and the other is the bioinformatics. So for the in vitro approach, what we have done is, as I mentioned, screen in parallel the panel of 8 medulloblastoma group 3 cell lines in parallel with the control cell lines. We have conduct and screening at the 10 micromolar. We have used a chemical library composed of 1280 compounds that cover more than 300 therapeutic targets, because we are interested in the targets that they are targeting. Okay, so what we have done is conduct this screening, and we have selected preliminary hits based on the activity and also on the preliminary in vitro therapeutic index. And what's this? This is that we select the hits, those that do exhibit more than 20 activity against the medulloblastoma cell lines, but we want at least 5 times more activity in the medulloblastoma cell lines versus the control cell lines. So we only select those compounds that do exhibit the activity, but do not exhibit preliminary toxicity at least. So we selected 8 hits that do target, say, 6 different targets, and we conduct the dose response curves, confirmation assays, and then we combine those hits in pairs. So we perform dose matrix of 8 rates, and we calculate the combination index with the Bliss and also THIP model. Okay, so at the end of this process, we select 3 synergistic combinations. And as we are aware that this chemical library is not so extensive in the sense of chemical space, we wanted to enrich our approach with this in silico approach. Okay, so what we have done is conduct this bioinformatic analysis to identify the genetic vulnerabilities and drug sensitivities in medulloblastoma cell lines. We have enriched this data that we have obtained from public domain with our results from the 8 hits, and with this we have we have identified novel and specific synthetic lethal interactions for medulloblastoma group 3. So here I show you just representative results for the experimental approach. So here you can see how we select the hits. So here you have the medulloblastoma, the screening of the chemical library against one of the medulloblastoma cell lines. As you see here, the set prime is 0.8, so really robust. And here you have the representative control cell line. So we select those compounds that do have activity against the medulloblastoma, but do not exhibit any activity against the control cell line. Here you can see the confirmation experiments that we conduct with these preliminary hits. So as you see here, I mean, here are this is the panel of 8 medulloblastoma cell lines, and what's more important is this blue is the BG, so it's the control cell line. So this is a representative selected hits. As you see here you have the 8 curves for medulloblastoma cells. Okay, and here you see the results for the control cell line. As you see, there is, it has no activity against the control cell line, and it do exhibit activity in the medulloblastoma cell lines. Here you have a representative non-selected hit where you can observe the blue line, that is the control cell line, is just the same as the medulloblastoma results. Okay, so we discard this kind of hits. Here you see the dose response curves. We perform them with 14 different concentration points, and here you see a representative selected hits. So you see here the 8 curves, against the 8 medulloblastoma control cell line. So no activity again against the control cell line. And here you see a representative non-selected hit where the blue line is overlapping with the medulloblastoma cell line. So this hit was discarded. Also, we check for caspase activity, so we are sure that we induce caspase. And here I show you a representative results of the synergistic combination. This is a matrix, okay? And this is the bioinformatic approach. As I mentioned, we wanted to expand our experimental approach with bioinformatics, and we integrate the genotypic profiling data with large-scale loss-of-function genetic screens to identify those genes, the target genes, whose function is really essential in the presence of certain medulloblastoma mutation that are the driver genes. So here we obtain the driver genes specific for medulloblastoma group 3, and we look for the druggable essential genes in the map, and also enriching the results with the analysis of our 8 hits. So at the end of the process, we identify novel dependencies specific for medulloblastoma group 3. Okay, so now from this analysis, what we have done is select our first synthetic lethal pair candidate specific for medulloblastoma group 3, while we are intensively validating the rest of the selected pairs. Is composed of a driver gene that is mutationally activated, and we have the target gene that is a proto-oncogene. What is really nice is that this driver gene is one of the most commonly altered genes in medulloblastoma and also in other solid tumors. So for future indication, this is really nice. And also, many therapies with this driver gene are using this driver gene as a predictive biomarker. So this is really nice because we can have also our biomarker for patient for selecting the proper patients for this therapy. Okay, and regarding the target gene, it's overexpressed or amplified in really different cancers. Okay, so it's really a nice synthetic lethal pair. Here I will show you representative results that we have obtained in the lab. So what we are going to do is that what we call target 1 is the driver gene, and target 2 is the target gene. Okay, so here what I see is the synergism of the combination of 1 inhibitor for target 1 and 3 different inhibitors for target 2. Okay, so here we have the synergism of this inhibitor A plus inhibitor 1 for target 2. Here, the inhibitor A for target 1 and inhibitor 2 for target 2. And here, inhibitor A plus inhibitor 3 for target 2. Okay? As you see here on the left, it's always the 24 hours, and here is 48 hours. So it's the same. And here you see in red, okay, is the target 1 inhibitor, and the blue one is target 2 inhibitors. So this is for inhibitor 1, inhibitor 2, and inhibitor 3. And in purple, you see the combination. So as you can see here, you observe really nice curves for the inhibitor for target 1, and also inhibitor for target here, inhibitor for target 2. And when we combine both inhibitors, you see that there's a shift on the to the left of the combination. So that means that really we can reduce the concentration of both drugs in the combination really, really a lot. I mean, and this is how we express. This is the CYP score, that it's 20. And just for you to know, is when a combination is antagonistic, you have a CYP less than -10, and if it's additive, is between -10 and +10. And for a synergistic, you have a CYP score above 10. So here you see that it's 20 at 24 hours, and here we have a CYP score of 19 at 48 hours. So this is inhibitor A plus inhibitor 1, and here we have inhibitor A plus inhibitor 2. You see the same pattern. I mean, this is inhibitor for target 1, inhibitor for target 2, and then this is the combination. And it's really synergistic, as you see, looking at the CYP score, okay? And here on the right, we have the combination of this inhibitor A, the same as on the left, but with the inhibitor 3. And you see that here at 24 hours, we don't have a synergism, but we have an additive effect. And at 48 hours, we still have an additive effect, but the CYP score is getting closer to the synergisms, okay? And the difference between those inhibitors 1, 2, and 3 is all of them share hydrophobic interaction features, but this inhibitor 3 do have a different core scaffold, okay? So we see that those combinations are really highly synergistic, and this is additive, okay? We have tested, here I present also representative results where you can see a general toxicity model in VJ, and you can see also the hepatotoxicity model. I only present the 48 hours because I think that it's more relevant. And as you can see here, this is up to 100 micromolar for the inhibitor 1, I mean A, and up to 10 micromolar for inhibitor 1. And as you see, there is no toxicity in any of the tested models. And I want to show you these results for a different combination, that is with the inhibitor B for target 1 and for the inhibitor 3 for target 2. And this is important because I want to stress the relevance of setting up really translational models that could anticipate the potential problems that we will face in the in vivo phase, okay? So here you see this combination, as always, this is for target 1, this is the curve for target 2, and this is the combination. Of course, you always see an additivity, so that's really nice, but not really a lot of improvement as we saw before. But if we move into the toxicity results, here I show you this general toxicity model with the PJ, 2 different neurotoxicity models. This is neurons differentiated from the lumens cell lines, and this is from the neural stem cells. And here you have the hepatotoxicity model in the HepG2 model. As you can see, here the scale is different from the previous one. Here, target 1 goes only up to 1 micromolar, okay? And you can see that single compounds and also the combination is getting really toxic. So this combination results toxic in all the tested models, okay? And here I want also to show you the results from this combination in our 3D models, okay? So activity and toxicity model. Here we start with a matrix of 3 plus 3. So you can see here that we have the 2 independent curves for both inhibitors, and here you see the curve for the combination, okay? And it's synergistic. But when we perform the whole curve with 8 concentrations for each, then we see that the combination is additive. And what's more important for us is that when we test this combination in our 3D neurons model, then we see that we have the same toxicity, neurotoxicity, in the same range as the activity. So this combination, it's additive, and it's also neurotoxic for, and it's also we just confirmed that we induce apoptosis, but okay. But if we go to our subcutaneous model, then what we see is that here you see the vehicle. This is the inhibitor B, inhibitor 3. Here you see it in purple, and in green you see the combination. And what we can observe that using this combination of two compounds, we do observe a significant inhibition of tumor growth. But this combination was really toxic for the mice. So as you see, the combination has only 4 mice left. So it's active, but it's toxic. And this is really in accordance with what we observe in our in vitro models. Okay, this is just a summary of the in vitro results with the different combinations that I have just shown you. So we observe inhibition in our medulloblastoma panel of cell lines. We see synergistic combinations for these 2 inhibitors. This third inhibitor that has a different chemical scaffold, close to synergy also. But what is more important is that the combination is synergistic or additive, but we do observe neurotoxicity and hepatotoxicity in our in vitro model. And we have also confirmed the downstream targets for this combination, and we do have our in vivo POC, so the proof of concept. So it inhibits the tumor growth. So also now for this synthetic lethal pair, our next step is the rational design of a dual inhibitor that will block these 2 targets. So we have conducted a structural analysis for this synthetic lethal pair. And both targets, the nice points of these targets is that both do have FDA-approved drugs, both selective and non-selective, and we have crystal structures for the protein ligand complex. We have really well characterized the binding site. We have Kiss-Ray and mutational studies, and we know the specific key binding features that will allow us to develop specific inhibitors. And what it's really nice is that both inhibitors do have a dual inhibitor. I mean, both of the targets separately do have dual inhibitors with the same partner. So target 1 with one partner and target 2 with the same partner, okay? So what we are going to do is to conduct a rational design of the dual inhibitor, and we will conduct a virtual screening. So this confirms us that it's feasible to design dual inhibitors. I'm sorry because I mean, you cannot see, but this has been done in collaboration with Nostrum Biodiscovery in Barcelona, okay? So just as a summary, what we want to stress is that we have developed a highly predictive in vitro platform that really anticipates in vivo activity and toxicity. The commercial inhibitors used as tool compounds do inhibit medulloblastoma group 3 cell survival in a dose-dependent manner, and the combination of both targets results synergistic or additive for killing medulloblastoma group 3 cells in 2D and 3D models. The combination does not exhibit general toxicity, neurotoxicity, or hepatotoxicity, except for the combination of inhibitor B for target 1 and inhibitor 3 for target 2. The combination kills medulloblastoma group 3 cells through cascade activation, and the combination significantly inhibits tumor growth in our xenografts. And structural analysis of target 1 and 2 reveals that it's feasible to design dual inhibitors for both